vignettes at the end about other salamanders, such as this Northwestern salamander right here. And I wanted to also add that the human component of this is not lost on me. I took a class forever ago now as an undergraduate called the Natural History of Oregon. It was a year-long class from Kenneth Walker. We called him Doc. And when I took this class, he kept telling these stories about Butch Brodie, and he'd tell these stories as though they'd happened yesterday. Come to find out, Butch Brodie had been there at Western Oregon. Back then, it was Oregon College of Education, 20 years before I showed up, 20 years. And Doc was still telling these stories. And I went, wow, who is this guy, you know? Well, it turns out he is the pioneer and kind of a grandfather. He'd probably, if he were listening to this, he'd resent it, but the grandfather really of anti-predator mechanisms, especially in salamanders, and was the first one to really start studying this business of toxicity in rough-skinned newts. So I went on from there and went on to graduate school and finally landed in a postdoc at Washington University. And I wrote Butch wrote him a letter and sent it. He was then at the University of Texas at Arlington. And he wrote back, and what I needed was tissues to do a big phylogenetic study of newts, the whole family that newts are found in, the Salamandridae. And I told him this story, you know, that he and I had both come from this common place, and we both started out being afraid of snakes, and we'd learned not to be afraid of snakes because of this class. And then later on, you know, how could I possibly have known that I'd end up in a laboratory right next door to Doc Walker's daughter, Charlene, who he talked about also during class, and who was then in the business of developing novel ways to clone zebrafish. And how could I possibly have known that Charlene would be the primary, the senior research associate with Chuck Kimmel? And how could I possibly have known that Chuck was on the board of the Natural History Society and that I would end up being president of that society and end up doing all of these things and involve this convergence of all these people? It just blows me away. I'm still just fascinated, and I had to take a couple of minutes to tell that story. So I always like to start out with a little bit of Amphibians 101. So you need to know a couple of things. And so what I always ask a class is, how would you describe an amphibian skin? And you're all muted, so you can't answer that. So I'm going to answer it for you. It's moist, right? It's moist almost always. Newts are a little drier, but most of them are very moist. They're slimy. They really are. Snakes are not slimy, but salamanders certainly are. And some of them are so slimy, they can produce enough slime to actually stick the head of a predatory garter snake to the ground. They're amazing. And they're definitely on the cool side, and that's because their thermal optima are usually, their preferred body temperatures are on the cool side. So there's a cross section of an amphibian skin for you to go through here, and I'm going to try this cursor. So this is actually a frog, and right here's a blood vessel. That's kind of important. Keep that in mind. This big thing is kind of the star of the show. That's a poison gland. Another word for name for that is the granular gland. And then here are some chromatophores. Those are the cells that give the skin whatever pigments it might happen to have. And then also we have mucus glands here and here. And most of these things have openings to the surface that don't show up in this cross section. The other interesting thing about amphibian skin is this very thin epidermis. Their epidermis is very thin compared to yours. And there's a really thin layer of keratin that goes around the surface of the skin. So what's interesting about that, and what we just described there then, is I'm going to try and move this out of the way. So we have this very thin epidermis with this thin layer of keratin, that stuff that your fingernails are made out of. It's a poor barrier to water loss. So amphibians dry out really easily. It's also an excellent organ for water gain as well, so it runs both ways. It's an excellent organ for oxygen exchange and exchange of carbon dioxide, because these blood vessels right here, these capillaries, actually run right up to the surface. And so any oxygen that's diffusing in there is easily accumulated in the circulatory system. The really cool thing is this dermis down here, this thick, complex dermis with all these amazing glands. They make pheromones for breeding. They make mucus for sliminess, and they make poisons. That's where the toxicity derives from. So how would you describe a reptile skin? Well, it's dry, right? They're all pretty dry. Scaly, definitely. There aren't any scales in amphibians. And it's often warm because the thermal optimum of a reptile, their preferred body temperature, tends to be higher than most amphibians. So just in contrast then, here's a cross-section of a snake skin. You have this much thicker layer of keratin. That's one big difference between amphibians and reptiles. And then the rest of the epidermis right here. Then the dermis is boring. There's nothing going on there. It's just pretty homogeneous, right? No really cool specialized cells like you see in amphibians. So you have this really excellent barrier to water loss. Doesn't exchange gases though, so they pretty much always have to respire through lungs. Amphibians are so good at getting oxygen through their skin that many of them, salamanders in particular, a whole huge family, have de-evolved their lungs. And you have just really very few specialized cells. So there you go. And there's a lot to tell other than that to tell as well. And there is the keratin, really, the shed keratinized layer, that right there, of a common garter snake that you might find out at Mount Pisgah or even in your backyard here in town. All right. So, toxic tail number 1, rough-skinned newt. I got to tell you, though, this whole talk is what other people did, almost all of it. I'm going to give you 2 things that I've actually done. One of them was, again, thanks to Butch Brody's help, providing tissues for this big study. Way back in 1995, I published a phylogeny, an evolutionary tree of the family Salamandridae. And so people always ask me, what's the difference between a newt and a salamander? And the answer is all newts are salamanders, but not all salamanders are newts. Okay, it's a hierarchical thing, right? So here are all of the newts right here on this part of the tree. And the other thing we found is that our rough-skinned newt down here is the closest living relative to the red-spotted newt in the eastern United States. Thankfully, since 1995, other people have taken up this question. Our tools were very limited back then. And by and large, a lot of that tree's been pretty well substantiated by other studies. So I feel good about that. And the other thing I feel really good about is a project I've been doing with Nearby Nature, and they call it Wow Newts. It's the Newt Quest. And so for 5 years now, I've been getting together with kids and their adults out at Tugman Park, and we talk about newts, and then we go out and count every newt in the creek. And it's great because the creek, that section of creek is really short. And then the city of Eugene, bless them, did a reclamation project where they ran the creek water around a really toxic dump that was polluting that creek, to the point that when I first came to the neighborhood, there were signs in the water telling you to stay out of it. It was so polluted. So they ran the water around that dump and introduced some meanders and riparian vegetation down below. And the amphibians are coming back. There are chorus frogs in there, and there are newts, and we count them. And I'm happy to say that they have been on the increase for 4 years. We started out with about 30 some, and we topped out 4 years ago with about 92, I think, newts in the creek. And we pretend to count them all. We count them on the same day every year, usually always in late April. Last year, we had a little bit of drop off because of COVID, and we got postponed for a couple of weeks. And I think it was really because it was just the adults that were counting the newts and not the kids that we didn't have so many. But I love that. I love that project. Great citizen science and great for the kids. All right, that's all I've done. The rest of it's other people's work. So if you don't like to talk, all the references are in there. You can go find them and tell them about it. So Tarika, I like to call them deadly, the rough-skinned newt, deadly but not dangerous. What do you mean by that, right? Well, there's a picture of our rough-skinned newt. that bumpiness is where they get their name, granulosa. Each one of those bumps is a collection of granular glands, which is a collection of toxin, I'll just say sequestering for now, cells on the skin. They're toxic throughout. All that entire newt is toxic, but they are especially poisonous in the skin, and I'll explain more about why that is later on. I like to ask people, this is the animal that people mention most often when you talk about salamanders. It's the one they see, right? And the question is, well, why is that the one you see? Well, the answer is because they can be out marching around in the middle of the day because basically nothing can really eat them. Here is a newt that this animal's been in the pond for a while. It's gotten a lot more smooth and slimy looking, and they actually kind of get some webbing starting to redevelop. It isn't really webbing, but the toes kind of swell and they get a blade-like tail. Some people call that reverse metamorphosis. This one's showing an Unken reflex. That's just a way of showing off this orange on the tail. They can even arch the chin up, show off all that orange and say, hey, eat me if you want, but it's going to be bad news for you. So question, what happens if you pick up a rough-skinned newt with your bare hands? The answer is nothing. I've been picking up rough-skinned newts with my bare hands since I was about 6 years old, and there's just not a problem with it. I read it all the time online. Oh, they're toxic. Don't touch, don't touch. Go ahead and touch them. It's fine, all right? You might not want to be rubbing your eyes afterwards, but you can pick them up. It's really okay. We wouldn't have a single kid alive in my neighborhood if they were that poisonous. The kids go out and play with them all the time out at the park. What happens if you eat a rough-skinned newt? That answer is pretty straightforward too. You die. You absolutely do, okay? You're going to be a goner, all right? And so really the thrust of this story is why. It's right, you die, okay? And the question, or the answer really lies in the fact that a lot of rough-skinned newts, but not all of them, have enough tetrodotoxin. And from now on, I'm going to refer to tetrodotoxin as TTX, because that has fewer syllables than that. There's enough toxin in those animals to kill maybe anywhere from 10 to 20 adult humans. They're very, very toxic. They're deadly toxic, but again, not dangerous. If you don't believe it, here's a medical case from that obscure journal from the American Medical Association. Not exactly right. This is a 1980 or 1981 case a case from 1979 that they published in 1981. 29-year-old man, sorry, drank 150 milliliters of whiskey at 11 a.m. Slow day, I guess, back in 1979. By 6 p.m., somebody dared him to swallow a newt, an adult newt. That's 20 centimeters. That's a pretty good size animal. Within 10 minutes, he said his lips were tingling. In 2 hours, he was complaining of numbness and weakness, and he stated that he thought he was going to die, but he refused to be transported to a hospital. They left him alone for 15 minutes. He went into full cardiopulmonary arrest. So the only thing that he really did that he had nailed in that whole evening, at least for himself, was the fact that he was going to die, and he did. He was a goner, okay? So it happens. There really aren't, in fact, very few documented cases of newt poisoning, but there are a lot of anecdotal stories out there. So the other amazing thing about that is that TTX doesn't cross that blood-brain boundary. So you get to experience the whole paralytic experience, right? You get to have it all. You're conscious for the whole thing. So, I mean, if you were thinking that the election was going to go bad and that was going to be just the end of it for you, and you were going to finish by eating a newt and being done with it, I don't think so. There just have to be better ways to do yourself in than that. I wouldn't recommend it. So how does it kill you? Well, great question, There's our amphibian skin again. And there's our granular gland right there. And that, again, is where all the action is with respect to these rough-skinned newts. Most of that tetrodotoxin is sequestered in those granular glands, and most of that happens to be in the skin. Although, as I said, they are toxic throughout their bodies, and that'll come up later in the talk as well. So how's it work? Well, it's a neurotoxin. Okay, it's a nerve toxin. And what it does is it causes impulses along a nerve cell to stop. You can't transmit a nerve impulse. So here's a nerve. There are some funny little fingery things out there called dendrites at the end. They connect maybe with another nerve cell. This particular nerve, here's the cell body going along here. And this one, importantly, actually directly innervates a muscle fiber. So that would be a skeletal muscle fiber. Then that would cause a contraction of that muscle fiber. How's that happen? Well, when the nerve is resting, okay, there's a whole lot of potassium ion inside the nerve cell, and there's a lot of and potassium ions are negatively charged. There's a lot of sodium ions outside the nerve cell, and they're positively charged. So there's this charge differential on either side of the cell membrane, which would be right here, okay? So when that nerve is stimulated, potassium goes out, sodium goes in. They call that depolarization. And there's this wave that moves along the body of the nerve cell. And in this case, would cause a muscle fiber to fire. So what does that have to do with TTX? Well, here's another diagram where we've just taken a bit of this nerve cell body and expanded it for you. And it's not voodoo that sodium and potassium can move in and out of that nerve cell. There are actually ion channels set up to do that job, all right? And one of them is called a voltage-gated sodium channel, and there's one right there, and they even give you sodium with some O8, an oxygen, and a couple of hydrogens stuck on it. And notice it's small and that's big, and it can flow through when it's allowed to do so when it's stimulated. And I don't go into that, but so this is how it's supposed to function. The sodium can flow from outside the nerve cell to the inside here and depolarize that thing, and that's critical in sending that impulse along the cell body. What happens? Well, enter our molecule, Tetrodotoxin. I actually only showed that for John Carter, and because John's a real chemist, and my chemistry background is almost as bad as my math background. Like, I stink at chemistry, okay? But I like to put these fancy diagrams up there just to make it seem like I know what I'm doing. The only point here is that there are parts of that molecule that can bind to that ion channel, okay? And so enter a molecule of tetrodotoxin. That thing gets stuck in there like a soccer ball would be jammed in a sewer pipe, all right? It blocks that channel. And when it blocks the channel, you don't get those changes that have to happen in positive, negative charge inside and outside the cell body, and you can't propagate an action potential. That's how it kills you. You forget to breathe and other fairly important functions like that, all right? You go into full cardiopulmonary arrest in the worst of cases. All right, that's how it happens. Turns out there are animals that are capable of preying on a rough-skinned newt. And, drum roll, they're garter snakes, okay? You see them all the time. We see common garter snakes and Northwestern garter snakes here in our neighborhood. You can see both of those species out at, say, for example, Mount Pisgah. Here's a common garter snake, also known as the red-spotted garter snake in our area, but there are other subspecies with different common names. Thamnophis sertalis, nice, beautiful checkerboard pattern, dorsal yellow stripe, and this spectacular red head. And I know that they can eat newts because I found this picture on the internet. And when you find a picture of anything on the internet, that proves it, right? Maybe not at all, okay? But so there's a common garter snake eating a rough-skinned newt. Spectacular. How how can this possibly happen? Like I just told you exactly how you can die and the fact that you actually would die. How does a garter snake pull that off? Well, back to our diagram here. There's our cell membrane with our ion channels and our tetrodotoxin right there. And remember, these are sodium channels here. There are changes that have occurred in the DNA of some garter snakes, and it turns out, you'll find out later, many other snakes, that in that sodium channel, that changes in the DNA that cause changes in the protein of the sodium channel that make these molecules of tetrodotoxin less able to bind. They'll still nudge up there, but they won't be stuck in there again like that soccer ball in a sewer pipe. And so the sodium can still get through that channel, and you can still propagate an action potential even when you've been inundated, your system's been inundated with tetrodotoxin. And so here, essentially what this is a 3D version of that ion channel right there. And they're showing these colored places, they're showing actual changes in amino acids that have happened in this protein right here. And so here's one, here's one, here's one, here's one, here's one. There are others that aren't shown, that those changes in amino acids change the charge on that part of the protein, and it makes the tetrodotoxin less able to bind. So that's cool. It's a great story. It turns out that there's this thing called an evolutionary arms race, and the classic example of this evolutionary arms race is the rough-skinned newt garter snake story. So it goes like this, and I'll just kind of walk you through this as best I can. Here's a picture of the range of the rough-skinned newt this is Vancouver Island right up here, going right down into California. This big white hole right there is Central Valley, the Central Valley of California. The only thing alive in the Central Valley is broccoli, as near as I can tell. So there's a big white hole there, okay? So the orange is areas where the rough-skinned newts are really toxic. They're very toxic. And down here to purple, where they really aren't very toxic at all, and green and purple and blue. So up here around Vancouver Island, they're not that toxic. They don't show a population over here in Idaho that you're going to see a little bit later that isn't toxic at all, okay? So that's prey toxicity. That's what they're talking about right there. Note, here we are in Oregon. Here we are in the Willamette Valley. Our newts are really, really toxic. Like they're some of the most poisonous newts known. It's just a hot spot. Here is something they're calling predator resistance. Predator means garter snake, and in this case, it means the common garter snake. That's what they were talking about in particular. Note that these colors tend to be very similar to one another. There's a high degree of predator resistance in areas like our area right here in the Willamette Valley relative to the toxicity in the newts, okay? And the reverse is also true. The snakes are far less resistant up in places like Vancouver Island where the newts aren't very toxic, okay? There's a huge variation in toxicity across the range of the newts. The idea is that the toxicity in the newts is being driven by garter snake predation, okay? So the newts become more toxic, the garter snakes become more resistant. They evolve, say, some more amino acid changes in those gated sodium channels to meet the newt's extra toxicity. The newts become even more toxic, and et cetera. Classic arms race, right? This slide's a little counterintuitive, but basically what they're trying to show here is the correlation between the garter snake toxicity and the newt's toxicity and the garter snake's resistance to the toxin. And the purple and green and blue areas are a high degree of concordance between the garter snake's resistance to the newt toxins, and the orange and red are discordance. And you can see a lot of concordance. There's a lot of matchup between those two things. So where rough-skinned newts are highly toxic, the garter snakes are highly resistant. where the rough-skinned newts have lower toxicity, the garter snakes generally have lower resistance, and there's this really great correlation between toxicity and resistance in the where the ranges of these animals overlap. And again, it's supposedly the idea is that the predators are driving the increase in toxicity in the newts, and the newts are matching that by upping their TTX in the skin primarily. And if none of that makes any sense to you, maybe this does. I love this cartoon. Yeah, there's our garter snake here, girded, right? And the newt getting ready to hurl a load of tetrodotoxin at its face. Actually, that cartoon isn't nearly as funny to me now in COVID times as it was a few months back. Everybody's wearing a mask now, you know. Okay, so let me complicate the story. Actually, I'm going to complicate it a lot. All right, we're going to dig deeper here. A group recently, that in 2016, then went about trying to understand. They studied the evolution of the resistance in these sodium channels. So I gave you a little bit of an intro really to the system back there when I showed you the picture of the 3D structure of the sodium channel and the amino acid changes that were happening in the protein of that channel to make the snakes more or less resistant to TTX. So McLaughlin et al. Really dove in. It turns out there isn't just one gene for the sodium channel. In amphibians, there are 6 of them, and there are multiple genes. They exist in a gene family. And this particular group went ahead and had a look at 3 different ones. And the way they did that, they gathered up 78 species of snakes, and they sequenced all of the sodium channel genes in these 78 species of snakes. And so by understanding the DNA sequence, then they could infer the amino acid sequence of the protein and talk about the evolution of the whole gene family, not just one particular sodium channel. And this is a tough one, but this is a phylogenetic tree showing the relationships of all those species of snakes. And they have some outgroups out here. There's a chicken and a turtle and a couple lizards, but the rest of these are snakes. And these are the different sodium channels. And in this case, they looked at 2 different domains, domain 3 and domain 4 of sodium channel 1.7, and then 1.6 and 1.4. And basically, it kind of works like this. There were some changes in sodium channel 1.7 and domain 3 and domain 4, and these were sodium channels that are expressed in sensory nerves. And those happened, you can look at the timeline up here and see somewhere around 278 million years ago, it would have been like that, the ancestor of all tetrapods. And here at about 170 million years ago, these 2 mutations. And some species only got 1, and some species got both. The blue lines indicate species on this tree that got both. Like, for example, turtles have both a change in domain 3 and domain 4 in 1.7. So that happened a long time ago. And the point is, those mutations were present in the ancestor of all snakes, and they conferred some mild resistance to TTX. The idea, though, is that they probably didn't even evolve for TTX resistance. They probably evolved for some other reason, like, for example, making the channel more reactive and more, say, making the nerve cell more permeable to sodium or something like that. They don't really know, but it didn't necessarily involve TTX resistance. Then you got another mutation here in sodium channel 1.6, and notice that all of the species that had these other 2 mutations then picked up this mutation in sodium channel 1.6, and those were in large myelinated nerves. What's a myelinated nerve? Well, that's a nerve with a sheath around it, okay, a sheath of myelin covering the nerve, and there are openings on the myelin that cause the action potential, the nerve impulse, to skip along the nerve and make it propagate a lot more rapidly, okay? So these 4 different snake lineages evolved this resistance in 1.6, and there's there's a coral snake. Let's see if I can get my cursor in there. Coral snakes are right there, and some other species. I don't even know some of these names. There's a hognose snake in there, and then here, then we're getting closer to home. This is the clade, the lineage that contains all of our garter snakes, okay? So 4 different times we had this change in sodium channel 1.6. Finally, in the red lineages, you get a change in this cumulative change. All 4 sodium channels have accumulated these mutations, and this last change then was they began about 12 million years ago, and here they are right here. Looks like 5 different snake lineages, and those were in skeletal muscle nerves. So if you think back to that diagram that I showed of the cartoon of the nerve cell, these are nerve cells that are in direct relationship to a muscle fiber, all right? And those came on only in lineages that already had these other previous mutations present, all right? And that actually conferred really extreme tetrodotoxin resistance. And so the cool thing is, I just put a red square around this clade right here. Those are our 5 Oregon garter snakes right there. Tertalis, Ordinoides, Atratus, Couchi, and elegans correspond to the common garter snake, the Oregon garter snake, Sierra garter snake, Northwestern garter snakes, which we have here in town, Western terrestrial garter snakes you can find even out at Fern Ridge or out in the Eugene wetlands. And so you have this cumulative evolution of tetrodotoxin resistance. And finally, in these 5 species of garter snakes, 3 of them are absolute champions, the common Oregon and Sierra garter snakes, when it comes to TTX resistance. And the other 2 are no slouches either, Northwesterns and Western terrestrial garter snakes, okay? Awesome. We have more champions in Oregon than just football teams, just saying. So that's amazing. That's an amazing story, all right? But there's this question, where do the newts get the stuff? Where do they get TTX? It turns out that as much as that system has been studied by Edmund Brody Jr. and his son Edmund Brody III, okay, they're on all those papers, this father-son duo, and a bunch of other people, like collaborators that they brought on. They have studied this thing to the nth degree, but still nobody has really determined where the TTX comes from in the newts. So there's been this debate between exogenous sources, that is, the newts get the TTX from someplace outside of them, right? Or endogenous sources, meaning they produce their own tetrodotoxin. For rough-skinned newts, there's been a circumstantial case made for both of those, and it looks kind of like this in terms of them acquiring the TTX from outside of themselves, exogenous sources. We know of a lot of species that have bacterial symbionts, like pufferfish, crabs, horseshoe crabs, some snails, blue-ringed octopus. They all get their tetrodotoxin from bacteria, all right? Some animals get their TTX from diet, and especially a recent study now that the Brodies were also on, caddisfly larvae eat newt eggs, and they have mutations in their sodium channels that make them resistant to tetrodotoxin, and the tetrodotoxin that's present in the newt eggs, they can tolerate that. So they actually become toxic themselves. And then finally, there's just no evidence for a biosynthetic pathway that makes this complex molecule, tetrodotoxin. On the other side, the endogenous side of the story, there's been no evidence of TTX producing bacteria in the newts, and that was based on some molecular genetics and PCR results back in 2004 when the field was still relatively young. They've also found that captive newts stay toxic with no dietary tetrodotoxin. So they don't have to eat it, clearly. And they've also noticed that if you get a newt to excrete a bunch of tetrodotoxin with electrical stimulation, they actually will regain their toxicity, also suggesting that they're making it themselves somehow, that they're getting it from an endogenous source. All right, enter Patrick Bailey. I love this, and this is the title of this paper that just came out in April, just this year. The skin microbiome facilitates adaptive tetrodotoxin production in poisonous newts. And I went, wow, I love this story. This guy, he's at Michigan State, graduate student. He's pretty much totally outside the fray, but he just thinks, well, he kind of wants to look into this toxicity issue in newts. And there was someone there, faculty there at Michigan State that was interested in talking to him about that and had been studying the system themselves. And so he went after it. And in fact, he was so afraid, so he tried to start culturing bacteria from newt skin, all right? Well, can you imagine trying to come up with an agar plate that somehow represents the environment of a newt skin? It'd be an extraordinarily difficult thing to do, and it was. And he was getting scared. Like, I got my dissertation tied up in this, and so he also did another thing where he sequenced all six. Well, he sequenced 5 out of the 6 sodium channels that newts possess. Okay, so he finally, but he pulled it off. It's just a great story. I love it. This is a scanning electron micrograph, 2 panels right here. This one shows a bunch of bacteria on the skin. Why nobody decided to take a picture of them sooner, I don't know, but there they are. There definitely are bacteria, but you don't know what they're doing. Amphibians are loaded with bacteria. They're also loaded with all kinds of antibacterial agents in their skin. And then this one, this top panel right here, I drew a red circle around that. That is the opening to a granular gland on a newt skin. Okay, that's the pore on one of those bumps. Okay, or well, actually one of the glands in one of the bumps on a newt skin, and a bunch of bacteria piled up inside that pore. Well, so what? But what he did was he ended up looking at the skin bacteria, being able to culture these things, learned how to culture them. And he looked at the difference in populations between these toxic populations from Oregon and a non-toxic population from Idaho. And he found that, in fact, he could identify 4 different tetrodotoxin bacterial strains, 4 different genera. There are probably more species than that within each of those genera, but definitely 4 genera of TTX bacteria. And he found that in the toxic newts, the skin was far less diverse in terms of its bacterial complexity. So it wasn't as diverse an ecosystem, if you want to think about it that way, but it was highly enriched for Pseudomonas, which was one of these TTX producing bacteria compared to the non-toxic newts. And those results were highly significant. On top of that, he found out that when he got a hold of all the sequences of all 6 genes, he sequenced 5 of them. One of them had already been sequenced, but he had all 6 genes, and he found that he could identify protein changes in the sodium channels of all of those genes that conferred high physiological resistance to tetrodotoxin. So he solved a bunch of mysteries right there, okay? One of them was, how are newts toxic without being toxic to themselves? Because in fact, you can find tetrodotoxin in all the tissues of a newt, okay? Well, it turns out all those genes are expressed in all those tissues, and they're all resistant to the poison. They're one of them, okay? Why is there tetrodotoxin in the eggs of a newt? Well, the mother gave it to them. Well, she did, but she probably actually gave it to them through her ovaries. She handed off a load of tetrodotoxin producing bacteria, probably, or maybe through the cloaca when the eggs were extruded out of the cloaca. That's the heritage that was handed to these to these eggs, which of course the caddisfly are eating, and the caddisfly are resistant to tetrodotoxin. Incidentally, if you throw a bunch of other aquatic predators in a jar with a bunch of newt eggs, guess who eats the newt eggs? Only the caddisflies. Dragonflies won't touch them. Predaceous diving beetles won't touch them. Nobody else will touch them. Only the caddisfly larvae will eat newt eggs. So this is amazing, I thought. And it just, this guy's worth $1 million in the future in terms of research questions. He's off doing a postdoc at Harvard in molecular biology. And so you can start asking these questions like, well, how is the microbiota, how do the newts regulate the microbiota of their skin? Because there's this obvious correlation between the snake resistance and the newt toxicity, and there's this huge variation. And so can it really be true, and it certainly could be true, that the newts are actually regulating their bacteria on their skin somehow, maybe by altering the environment with peptide secretions of various sorts. Who really knows that? But and then finally, how does the microbiota alter the cell composition of those granular glands? Because again, that whole research group that included both of the Brodies have discovered that the granular glands of, say, those animals that are low in resistance have far less of a particular cell type contained within them than the granular glands of the really toxic animals. So there's this obvious morphological difference, and it seems like it's being driven by the bacteria that occupy the skin. It's amazing. I'm glad it's him and not me. I'm retired, but it's amazing. All right, so we talked about newts, and I showed you granular glands and all kinds of cool stuff, and bacteria and snakes that eat them and drive increases in toxicity that somehow happen because a change in the bacterial population. And the newts are responding to this by developing these behavioral responses to advertise their toxicity. It's an amazing system. It gets even messier. I mean, just a couple of little vignettes here. Turns out that if a snake eats a newt, they actually become toxic themselves. All right, so they pile up enough tetrodotoxin in their liver that it would actually severely impair any number of mammalian predators and probably kill any number of avian predators for about a month. And that just one newt. If they eat 3 or 4 of them, they're poisonous as hell for a while, okay? So that's interesting. You have a non-venomous snake becoming poisonous. It's not venomous, but it's becoming poisonous itself, which raises the possibility, like, what about those pretty red checkered spots and the yellow stripe on a common garter snake, you know? Maybe that's an advertisement that they're actually poisonous. Who even knows? It turns out also that if you look at snakes from California, when they're homozygous, meaning they're carrying 2 copies of that allele in that sodium channel 1.4 with these various mutations in it, if they carry both copies of that, they don't crawl as fast, okay, as animals that are either heterozygous or don't have that allele at all. They're much better at crawling. So it seems like there's a cost to the snakes to have that mutation that gives them resistance. That costs them in terms of their ability to crawl away and perhaps get away from a predator. And so it looks like this evolutionary trade-off that's going on in the system as well. So, you know, it's a mess, but it's a great mess. I love it. It's really, really fun. And they just keep finding out more amazing stuff. All right. Toxic tale number 2. This one won't be as long as the last one, and it can't be. So, will kissing that toad really make you feel like a prince? I mean, you may not turn into one, but at least you could feel like one, right? And I'm going to focus most of this discussion really around our western toad, Anaxyrus boreas. Some of you may have learned it or know it as Bufo boreas. The toads have been split out into a number of genera for reasons we don't need to talk about. Bufo boreas had a nice ring to it. I really miss it, but it's just the way it is in taxonomy. We got to keep up, all right? So they're cool. And it turns out that they have a bunch of granular glands too, and they're piled up in these big structures here. They're all over the skin actually, but they're piled up especially in these structures on the side of the head. Those are called peritoid glands. They're more like a glandular structure, okay? Then there are granular glands inside those with cells inside those that are making these various cool chemicals, all right? And you can recognize them in our part of the world because of this dorsal yellow stripe. So this is a true story. I had a student years ago, came up to me and she said, I want to do an extraction from a Western toad skin and smoke it. And then I want to keep a journal of my experiences. And my answer, because I'm a calm, demure, patient individual, want something like this. Are you crazy? You know? And then I had to remember to be respectful, and because she clearly was in her own way, you know, just no, you can't do that, right? There's no possibility that a project like that was ever going to happen in my class. But I thought it was amazing that she would even ask. Okay, so what about that? I mean, she didn't cook this thing up in a vacuum. She really didn't. She actually knew a little bit of stuff before she had come to me with this idea about toads and toad toxins, all right? And there actually are a couple of species, and at least a couple of them. These are the only two that have been studied, or I should say maybe experienced in any detail. One of them is a Colorado river toad right over here, Encilius alvarius. And I think John Postlewaite's somewhere in the audience. John's actually seen these. He sent me pictures of them from the Colorado River trip. And big peritoid glands. That's a cool looking toad. It really is. And this thing, the cane toad, Rhinella marina, used to be known as Bufomarinus. That's a badass toad. That's a formidable animal. And in fact, they get to be about the size of a dinner plate. You know, they get big. I saw one in Costa Rica parked next to a column of army ants, just lapping up ants as they went by. It would just lunge, lap, you know. They're amazing. And they're also incredibly invasive. They've shown up down in Florida now, as has every other species of tropical organism that probably could be held in captivity that was then turned loose. They're in Florida. They're all over Australia. They're infamous, right, in Australia. So it turns out both of those species have a compound in their skin called bufotonin. And the reason I'm showing a little bit of its chemistry right there is this. So bufotonin is 5-HO-DMT, and there's a chemical structure. And again, I just put that up there to make you think I know something about chemistry. It's just cool. And besides, I can make the animation spin in little circles, and it's fun. Bufotonin is very similar to psilocin and to LSD. Both the hallucinogenic components of LSD and psilocybin mushrooms, all right? That's interesting. It also turns out that it binds to serotonin, hallucinogenic, I should say, serotonin receptors in the brain cells. And you all probably have heard of serotonin. It's one of your feel-good chemicals. It's the chemical that might get you through 2020. It's the chemical that, if it's not going to get you through 2020, you take drugs to inhibit the uptake of the stuff so you have enough in your system to get you through 2020, all right? Those are serotonin uptake inhibitors, all right? So you got this chemical that actually does the work of a good hallucinogen. And so you start hunting around online. This is the title of a paper. A single exhalation of vapor from dried toad secretion containing 5-mexi-NN blah, 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 in a naturalistic setting is related to sustained enhancement of satisfaction with life, mindfulness-related capacities, and a decrement of psychopathological symptoms. That's the title. Well, you know, who even needs to read the article? Stuff sounds amazing, right? Especially the decrement of psychopathological symptoms. I can totally relate to that. Here's something that Wade Davis wrote back in 1991, and you may have heard of Wade Davis. Shortly after inhalation, I experienced warm flushing sensations, a sense of wonder and well-being, strong auditory hallucinations, insect cicada sound that ran across my mind and linked my body to the earth. I was indoors, but there, the feeling of the earth, the desert soil passing through my fingers, the stars at midday, blah, blah, blah. I mean, wow, you know, and I honestly, you know, I read that and I thought, wow, if I could write like that, give me some of that stuff, you know. And I mean, besides Wade Davis, he's a famous dude now. He's written a bunch of books on ethnobotany and pharmacology, and he's an a cultural anthropologist now working up at University of British Columbia. He was here at the U of O some years back, and I missed him. I wish I had been able to take in his talk. But so that's pretty amazing, you know. So there's our Western toad, and just a reminder that of our amphibian skin right here, and the fact that all this action's happening right here in these poison glands, these granular glands, and also to tell you that they're found pretty much all over the state, except here in the Willamette Valley. They're of course most common up out in the Cascades, and a lot of you have been hiking in the high lakes, especially in late summer when the tadpoles are metamorphosing and you practically can't walk past a little lake without squishing toads under your feet. There are just jillions of them coming out of the water and migrating out into the terrestrial habitat. So, like, why not? You know, let the students smoke some toad skin. A little chemical stimulation, a little shamanic experience. What? Heck yeah. A little Wade Davis action. I mean, be a psychonaut or nut, depending on your point of view. Well, you know, what could possibly go wrong? Basically, a lot of things could go wrong. And so the thing you got to remember is that amazing skin and the toxicity that often evolves along with it, that did not happen to make you happy. That happened for reasons that don't have anything to do with you. And so, of course, it's a lot more complicated and not all exactly what you think. So there's this whole collection of things called bufotoxins, and there's a lot of chemicals in bufotoxin. But one of them is adrenaline. Well, a little bit of adrenaline is great, especially if you need to get away from a predator or give a talk, for example, to a natural history society. But too much of that stuff, it's going to cause a heart attack. All right. You don't want to overdose on adrenaline. Likewise, it's close relative noradrenaline. Little bit's good, too much, heart attack city. Then you get into some other nifty things. This is Bufogen. It's a lot like digitalis. Digitalis is the chemical that you find in foxglove. Digitalis is a known heart drug, and a lot of people need it and can use it to increase the strength of a weak heart contraction because it causes a buildup of calcium inside the heart cells. But I mean, the downside is if you don't regulate it properly and take it at very carefully monitored doses, it can actually cause ventricular fibrillation. Another one, Bufotonin, it's neat. It's an anti-cancer drug. Did you know that toad skin is full of anti-cancer drugs? But it also works like digitalis does. It can strengthen the heart contraction. Then at some point, your heart gets weak and then it just stops, all right? Bufotonin also has anti-cancer properties. These things are known, increasingly well-studied tumor suppressors. And what they do is they actually make the cells in the tumor increasingly prone to cell death, to apoptosis. But again, you know, you get these effects that are similar to digitalis. And finally, bufothioneine, it's another anti-cancer drug. And in that case, the cardiotoxicity isn't really documented. So there's this whole cocktail of things. So, you know, you look at that and you go, do you think your heart just might be at risk if you go and smoke that toad skin? Well, I mean, that's a duh, right? Of course it is. So I didn't know any of this when I aborted my student's project and we got her off onto something a little more reasonable, but I feel pretty good about having told her no now in retrospect. So yeah, you know, Colorado River toads do have high levels of bufotonin relative to the other cardiotoxins. Wade Davis has had a long and successful career since 1991 after smoking that stuff and having this incredible experience. He did mention that he did have an increase in heart rate in that experience, but you know, it worked out okay. And in fact, the toads are in some places having trouble because everybody wants to go out and smoke a damn toad, you know, and so it's actually threatening populations of some of these things. And you can even find websites of rehab facilities that are writing these online pieces about smoking and licking toads and trying to get people to just sort of steer clear of all that stuff. I mean, it's a big deal. And in fact, my student, with what she knew at the time, was correct in hypothesizing, even though she wouldn't have formalized it like this, that closely related species might have similar chemical profiles because there are evolutionary constraints on systems. And in fact, closely related species do tend to be really similar in many respects, and maybe also in chemistry. So we got to ask the question, right? How closely related is a Western toad to a Colorado River toad? And it turns out not very. So I'm going to close that chat panel. There's a phylogenetic tree of these two genera of toads, and I've drawn these big flashy orange arrows to show you there's Anaxyrus boreas, our Western toad right there. And then it's in a clade that's completely separated, not distantly related, but very separate from this clade right here that has the Colorado toad. This is Encilius alvarius right here. And in fact, the marine toad is separate from both of those. So they aren't really, in terms of toad phylogeny, very closely related to one another at all. And I'm not getting my slides to advance very well for some reason. Let's see, there we go. So, you know, proceed at your peril. And this Esther English article put it, I thought, very eloquently. The overall effect of toad licking can cause a person to have vivid hallucinations, a racing heart, and muscles too weak to carry their constantly vomiting body to the bathroom, let alone the hospital. I love the internet. You can find anything. So, a couple of other toxic tales. The Northwestern salamander, Ambisoma gracilli. My wife says they look like space monsters, and they kind of do. And part of the reason that they look like space monsters is, again, you might recognize this from what you saw on the toad picture. That's a peritoid gland with a bunch of granular glands concentrated there. And if you harass these animals, they'll do a kind of a head swat like that and swat at you with their tail. And if you look, you'll see this white stuff oozing out from around these, see if I can find my cursor again, these peritoid glands right here, and then also this glandular ridge along the tail. And in fact, that stuff is toxic. Brody Jr., Butch, studied that toxin in some detail, and also the granular glands in Northwestern salamanders. And but this is classic, right? So Butch is a real fan of getting to know his salamander toxins, and so he put a drop of that stuff on his tongue, and he said it caused drying, loss of touch, and a burning sensation that lasted for 15 minutes. You go, Butch. He's had other encounters with amphibian skin, some of which he's published, and others he has not for reasons I can't go into. So Here's our Oregon ensatina. That's a lungless salamander. And a way to recognize them is this, you can see it kind of right here, this constriction at the base of this round tail. Well, that tail has loaded with poison glands, and they also make a sticky, milky secretion. And then finally, we have poison showing up in lots of things with no tails at all. There's the Great Basin, spadefoot, and Spia intermontana. You can recognize them. They have vertical pupils. They're cuter than the dickens. I love these frogs. I think they're my favorite frog, I think, of any we have in Oregon. This hourglass, white hourglass pattern on the back, they're about like that as adults. And then you see these cornified spades on the hind feet, and those are actually functional. They'll burrow with their rear end, just start ooching down into the sand and disappear. They make skin secretions that cause sneezing, runny nose in humans, but that's all we know. I mean, these things haven't been studied at all. I might also add that they smell like peanuts. And I'm not the only one. It's been published that spadefoots have this peanut smell that might be related to those skin secretions. So there's a ton we don't know, an absolute ton we don't know. So I'm going to summarize this mess for you as best I quickly can. So I showed you that amphibian skin's really complex. It has a lot of different glands that do a lot of cool things, but some of those glands actually sequester. And notice I say sequester now, not produce, because especially in the case of the newts, the glands are being produced, the glands are sequestering toxins that are being produced by a bacterium, and also deliver those toxins. And they do that. They generally have a sheath of smooth muscle around them that can contract and squeeze that stuff out. Rough-skinned newts, a lot of them have lethal doses of TTX. Those are produced by skin bacteria. The newts are resistant to their own toxin because all their sodium channel genes have evolved resistance to the toxin. And garter snakes have evolved a lot of resistance to those toxic newts, but you have not, okay? So don't be going out and drinking a half a bottle of whiskey and taking some crazy dare. Just stick with the whiskey and you'll be all right. There are a few toads that can give you a pretty nice buzz, it sounds like to me, from all I've read. But most of them have this cocktail of skin secretions that are just loaded with cardiotoxins. And for my money, it's not really worth it to go messing around with that stuff. And besides, be nice to the toads and go find something else to smoke. It's easy. It's even legal now. So, and then a lot of amphibians have other toxins, and most of them have not been characterized really hardly at all. Not anything like what we've seen in the newts and the toads. And I think a lot of what's happening with the toads and the reason they're becoming so well characterized is because of the anti-cancer properties of a lot of those compounds in the toad skin. And this is my favorite, funniest thing Butch Brody ever told me. If you need to find out how an amphibian tastes, try to find somebody else to do it. He's been around a long time and he's learned a few things. You guys want a bedtime story? I think I have time. Do I have time, August? Be about 3 more minutes.
Yeah, go for it.
Yeah, sure. So I've been writing a whole series of things. I actually haven't been writing as much in retirement as I hoped I would. But one thing that I've done to keep myself going is I have a series of things. I've been posting them a lot on social media, and some of them are on my blog. You can find that on my website. This one actually is on my blog, on my website. They're called Facing Down the Apocalypse, and they go 1 through, I think I'm up to about 22 now, all right? This one's number 6, rough-skinned newts. There sure is a lot of sex at the end of the world as I thought I knew it. After a socially distant run on the chip trail, I finished with a sweaty stroll through the neighborhood park. pausing on a footbridge across the small creek. The air has become a breathing thing, ribs of willow and cottonwood exhaling, unfurling the riverine smell of April trout season. Afternoon sun angles out of an emerald eye sky, filling a clear pool where the creek pauses below the bridge. In sun-split water at the edge of the bridge, at the edge of the day, at the edge of everything, a gaggle of rough-skinned newts is absorbed with making more rough-skinned newts. Milk chocolate backs blend with the salt cobbles strewn on the bottom, but each is outlined with a trace of apocalyptic orange seeping out from belly and tail. Orange tells a tale of toxicity. There's enough nerve poison in the skin of an adult newt to kill 10 humans. They're poisonous, but not dangerous. Just don't eat one. A pink nightcrawler the size of my pinky hangs half swallowed from the mouth of one newt, an ambitious undertaking. A wad of 6 squirms in the submerged roots of a willow, 5 males vying for a single female. Others in the pool are paired off, males clasping the female from above, a swimming undulation of sex that could continue for 2 days. Imagine it, or don't. If the male fills the bill, female will use her reproductive opening to grab a tiny plug of sperm he leaves for her on the creek bottom. Afterwards, she'll glue her fertile eggs to submerged twigs and return to the terrestrial neighborhood. The males stay in the creek, hoping to mate again. Hope. Now there's an interesting end of the world word. I wonder if newts know the difference between hope and instinct. I sure don't. I'm not enlightened in newt consciousness, but it seems to me they simply show up in the creek looking for sex. 2 days of continuous sex, if that's what it takes to extend the future of newts. There's an intelligence in that. But human ancestors crawled out of the water a long time ago. And even though we still carry a bit of newt brain with us, the end of our world requires that we blink in the spring sunshine and bring forth a bigger brained version of love, a love that reaches wider, a love worthy of all that gray matter we carry around in our skulls, a smarter kind of love that rises above all that squirming, jostling, shouldering, pushing aside of others to procreate an economic system that has become toxic and dangerous. We're smarter than newts, right? I sure hope so. Thanks, folks. I had a blast.
Yay.
Thank you, Tom.
Thanks, Tom.
Thanks.
Thank
Let's see.
you, Tom. That was wonderful.
You're welcome. Let's see if I can get this to go.
You.
Ah, there we go. Sometimes cartoons are close to the truth. That one sure was. So I guess I'm going to go ahead and pull up the chat window for starts and see what showed up. Maybe I can just stop the share now and grab that window. Looks like there are some entries.
Quite a few.
Oh my goodness. That's overwhelming. Yikes. Oh. Okay. Lois, oh, you teach, yeah, natural history in Nevada. Yeah, you're gonna have to look hard to find salamanders in Nevada. That's a tough one. Rebecca's telling you that you can go to the website and find a form to join us, which I'd also love for all of you to do if you haven't already. And also, I think August, there's going to be a link to the talk, right? The recording there
On the website?
Yeah.
Yes, that'll happen once I get it up on YouTube, and I'll
Okay.
pass that link on to Tim.
And for those of you that are connected with me on social media, I'll put it up there too, but that doesn't get to everyone. So why the high discordance and toxicity in newts on the Central Coast? You know, I've looked at that hard, and the short answer is I don't know exactly, but if I squint hard at that diagram, it seems like it's one of those places where the newts, the snakes seem to be a lot more resistant than the newts are toxic. So this is from Lois. So that's a possibility. And they did make reference in their paper that there are some places where the snakes are quote unquote winning that arms race, meaning their resistance is outstripping the newt's toxicity.
That's very interesting, and I'm gonna go read that paper because I was totally enchanted by that little spot on the coast.
Yeah, yeah. And it has to do with I think some sampling error too, and how they assayed the toxicity. You know, you're going to get some error introduced that way as well. So let's see.
Of course, but it'll be fun.
Yeah, it really is. It's a cool paper, and the articles about the paper are as much fun as the paper. You know, they're really fun. Do other animals other than garter snakes eat newts and have resistance? So I don't know, probably not necessarily newts, okay, because those are our rough skin newts are a West Coast thing, but newts in general, there are a lot of newts and they're scattered worldwide, and I don't know exactly what might eat them and what wouldn't, but what we do know from that study on the evolution of sodium channels is that in fact, some toxicity, some resistance to toxicity has evolved in quite a few lineages, and it looks like it's evolved convergently. And so given that they only looked at 78 species of an infinitude of snake species, all right, it would seem reasonable to me that some snake along the way got it figured out, quote unquote, in an evolutionary sense. But I don't know the answer to your question, but only that it seems like a good, strong possibility that there would be other resistance evolved. Yeah, great horned owls, they would die. That's from Lois. And hi, Mary. Mary was one of my students. She survived. She didn't eat a newt. Have toxic newts been moved to non-toxic newt areas? And if so, I don't think so, but that would be a question for those folks. I don't think they've done those transplant experiments, but they certainly have demonstrated in no uncertain terms that that snakes that aren't resistant will definitely die if they eat a toxic newt. The other interesting thing about that story is that the newt skin, the newts all have resistance to tetrodotoxin. They have all those mutations and all those sodium channels that make them fully resistant to tetrodotoxin, even though they don't produce it in those populations where they aren't being driven apparently by snake predation. So, I mean, it's just a wild ride, you know. I mean, how does that happen with this bacterial intermediary? It's amazing. Hey, Abby, how you doing? Oh, what do the snakes eat in BC? Garter snakes will eat anything they can get in their mouth. Okay, so there are plenty of other things to eat, even though newts are fantastic. I mean, a big adult newt is a major meal for a garter snake. And given the fact that these animals are all ectotherms, meaning they're cold-blooded, quote unquote, their metabolic requirements are pretty low. So it's a big meal. But our students caught a big common garter snake out toward Mapleton one year, and it had this giant bulge in the abdomen, and I went, oh my goodness, you know. And so we kept the snake because we hang on to things for a little while so that everybody can see them, and we can talk about how to identify things and all that. And so we had it in a cotton bag, which is how you should always keep a snake. If you're going to keep a snake for a little while before you turn it loose, so please turn it loose exactly where you found it. And it irped up a half-grown bullfrog. And it was astounding. I mean, I couldn't believe that it got this thing in its mouth, you know. So they really are remarkable. They do feed on a lot of different animals. They feed on a lot of different amphibians. They love chorus frogs and other salamanders, all kinds of things. Licking toads. Do newts hibernate? They don't exactly hibernate, but they do go underground and they do pretty well shut down because, I mean, true hibernation is a whole host of physiological responses that happen mostly in mammalian systems, right? And newts aren't really doing that so much as just slowing way down, okay? Their heart rate becomes really, really low, and they require no food during that time, just no activity whatsoever, and they just ride it out underground, literally chilled out, okay? But yeah, you can find them almost any time of the year. People are already sending pictures to me of rough-skinned newts in amplexus. They're already starting to breed in a few places. But summer's not so good for them, even though they tend to be more resistant to desiccation, to drying out than other animals. It's not a great time to be newt watching, but pretty much the rest of the year. I don't know what Oregon's done to, this is from Abby. Oh, sorry, and that previous question was from Carla. Abby asks, does this mean the state of Oregon just decriminalized toad licking? Great question. Honestly, I don't care. It's still a bad idea. Right.
I was just wondering if it was some sort of caveat.
What was that?
I was wondering if it was some sort of caveat in the new law that was passed.
I don't know. I mean, if you don't die, it only lasts for 15 minutes, right? What's the big deal? So this is from Carla. Do you think someone should pick up garter snakes? Yeah, no problem. They bite like heck. You know that, right? And they mess all over you, and they're smelly animals, and they're not my flavor of the month, man, garter snakes. I pick them up, but mostly for my students. I had one drop blood on me a couple of years ago. But yeah, you can pick them up. It's no problem. And it really, if they bite you, you're not going to get any fatal dose of neurotoxin at all.
Yeah. And sorry, Tom, it's Carla. I didn't want to interrupt, but hi, everybody. Hi, Ruth Braymiller. It's Carla Herbert. But anyway, so the reason I asked Tom, and not to give my whole personal history, but I've been paralyzed twice from vaccines. And what it is, you know, the whole demyelination of the myelin sheath, you know, kind of a similar cell structure from, you know, the antigens, the antibodies, you know, they attack the myelin sheath. And so when we moved here in 97, I had a pre-employment vaccine, you know, because I'm a nurse. And so I was paralyzed, it took me a year to walk again. And so I mean, I love all these critters. I don't want to be stupid, you know, but
yeah.
I can't do vaccines. I have to, you know what I mean? I can't do rabies bites.
Yeah, you shouldn't be picking stuff up, really. And,
okay,
I mean, really, you know, because garter snakes, even though they're not technically venomous either, it turns out that a lot of snakes do have some low levels of toxin in them, and plus just bacterial issues and everything else. You shouldn't be. You're high risk. Don't pick them up.
Okay.
You don't have to. I won't make you.
But I could pick up newts because they don't bite.
Yeah, you could.
Okay, thanks.
Let's see. Okay, I'm gonna skip. You're welcome, Mary, and Carla. Stan, hi. How have you been? Are you in Eugene? Question for you.
No, not yet. I'm still stuck in New York.
Ah, okay.
Yeah, so it's getting to be bedtime.
So what you all need to know about Stan before he goes to bed is my first serious salamander hunt that really kind of got my career rolling. So it was with Jim Keyser. It was in actually the bio that John Carter wrote for Nature Trails. It was with Jim Keyser, who and what we were doing, we went off to Costa Rica for a couple of weeks with these guys, and we were getting salamanders for Stan's research, his dissertation research on chromosome evolution in Central American salamanders, and it got me rolling. And so, thank you for putting up with me.
That was a fun trip. I think about it a lot.
I know. Well, there's things he's not telling. Mute him right now.
We should go back and see if we can find your wallet.
Yeah. So, Sam, that's a great question. Yeah, right. I knew it. See, you should have muted him. Sam's asking if newts produce TTX via their skin bacteria, what are they using the poison glands for? And it turns out they're sequestering the toxin that the bacteria are producing inside those poison glands. That's what's going on right there. Yeah. So great question. Do we know the LD50? Somebody does for Tetrodotoxin. This is, I don't have the full name. M. Hertz.
Me.
Oh, hi there. Yeah, so do we know the LD50? I don't know. I'm really sorry you had to take organic chemistry too. I had to do that once.
Well, actually it was very nice. John Kino was an excellent prof.
Good.
I think he gave the number of 6 picograms so he wouldn't be snooping around his lab, because stuff is, as you know, quite poisonous.
Yeah.
And, you know, he was always afraid that we might dump it down the drain and poison the city of Eugene. But
yeah. So that brings up a really interesting point. You know, if you go to PubMed, which is a huge database for the medical literature, and you type in Tetrodotoxin and hit go, you're probably going to blow the hard drive up on your computer before you get all the results. You're going to get tens of thousands of publications that have used Tetrodotoxin in one way or another. And the reason for that is because it's become hugely experimentally important to studying healthy versus unhealthy nerve function, right? Because it's so good at what it does, you know? Yeah, great question. 6 picograms, wow. And then any other questions that didn't show up on the chat? Chuck, are you on? No? Anybody?
Ask a question he didn't want to.
Hey, Tom, it's Mary.
Mary, nice to see you. Actually, I can't see you, but
I know.
I like to hear your voice.
It's nice to see you too. Here, I'll take my camera off. I'm wondering, are they looking into other animals that carry these types of toxins on their skin as well, and into like the bacteria that are associated with them?
Yeah, actually, they're way ahead with other animals. Yeah. Okay. A lot of organisms are now known to harbor TTX producing bacteria. So like the most famous, right, is pufferfish. That's kind of where tetrodotoxin was discovered, right?
Yeah.
They get it from bacteria. Yeah, so that was a huge clue for this graduate student at Michigan State to forge ahead on this, even though supposedly it had been shown previously that there weren't any TTX bacteria on the skin of newts. The problem with that was the molecular techniques in 2004 are not the same as the molecular techniques in 2020. That's one of the reasons I had to retire. I couldn't keep up with it all, okay? And so they've just become a lot more sophisticated at being able to recognize genetically these bacteria. So he went ahead with it, you know, and kudos to him for taking that risk. But his huge clue was that these toxins were already known from bacteria and other organisms. Yeah.
Yeah.
Okay, thank you.
So you learn.
So is there sort of I'm muted. No, can you hear me?
I can, Chris, go ahead.
Okay. Is there a line between the nemocysts that are in aquatic things are produced by the animal, right?
Yeah.
Is there an evolutionary line where that crossed or did they eliminate the bacteria need, or?
So I have the short answer is I have no idea. The nematocysts, of course, are hugely toxic, you know, but a kind of a sophisticated way to form that up. They talk about horizontal gene transfer. And that was still one of the possibilities for animals that harbor the bacteria, right? We have, our genome is loaded with bits of stuff that didn't come from us, okay? I think, John, are you going to do that piece on viruses?
Yeah, I think I will.
Yeah, so we have all kinds of bits of viral DNA and everything else that have arrived in our genome by horizontal gene transfer. So we didn't get it necessarily from a descendant. It got sent directly to us somehow or another. So it's always a possibility. And it's a possibility for TTX producing genes as well. But there's no way, I don't think, that it could happen exactly unless there was prior resistance in the animal that acquired that genetic machinery. I don't see how you could do it. So anyway, I don't know the answer to your question, but it was fun talking for 2 more minutes.
All right, Tom, so what about the genomic resources for these newts and salamanders?
Oh, man.
Do we have any genomes available that we can poke around in, or should
Pete
we
knows
some?
Pete knows the answer to that question. He didn't know how much that bottle of beer cost the other night, but he knows the answer to that question. And so the major genetic model system is axolotls. And the difficulty with salamander genomes is they are the monsters from hell. They're huge, okay? And most of that genomic DNA, I'm not going to say it doesn't do anything. A lot of non-coding regions do a lot of really important things, but in fact, it separates the coding regions that are making things like voltage-gated sodium channels by huge expanses of genomic material, okay? So it becomes very difficult to go sequence the axolotl genome, and the axolotl is our genetic model system. But I think we got all the coding genes right. I think that's been done, the transcriptome, and we've got that all figured out. But you know, I started working in the lab, my first DNA experiments at the University of Kansas when I started goofing around with this stuff were with salamanders. And I'd extract some DNA from salamanders and do this little precipitation step. And it was like a bringing cumulus cloud in that microfuge tube, you know, it was awesome. And I went, wow. And so, but then I started doing some work on fishes and lizards, and I do the same precipitation step. And I'd look in there and go, fail. You know, there's no DNA in there. I can't see it. Well, there was plenty of it. I just couldn't see it. It's just that salamander genomes are monstrosities. So is that what you're searching for? Was that
We've got the time, Tom. We've got the time.
No, you don't actually. We're getting past bedtime. I read my bedtime story. I'm not going to sequence a newt genome, right? I retired.
Yeah, and Pete, would you make a student do that? Would you make Tom go back to work, sequence the genome, and then make a student assemble it?
I wouldn't even do that to you. I wouldn't do that to you, Abby.
Yeah, I mean the lizard I assembled was, the genome size was like 3 gigabases.
Yeah.
Axolotl was like 10 times that.
Yeah, it's a huge thing. Yeah, so a lot of you don't know, but Abby and a couple other graduate students through Pete and also Jason, who's here, we've all teamed up to sequence the genome of the sagebrush lizard, which lives out across the Intermountain West. And one of the talks that I'll probably have to do a talk in January, right, August? That was sort of a joke. But one of the talks I also have up my sleeve is this sequencing project and trying to look at what we really want to do is get some DNA out of a 12,000-year-old lizard mummy from Paisley Caves and match it up to that genome and do some cool things. But Abby was great, a great participant in that project, and now she's on to bigger, maybe not better, but bigger things.
Nothing is better than that.
You're going to have to come back and actually catch one of those animals, you know.
I know, I'll have to noose it with your class when we can finally do it safely.
Okay.
What Tom didn't tell you is that he secured the funding for that project solely.
All $300.
So I'm waiting for the salamander project, Tom. I'm waiting for it.
Well, multiply 300 by 10. That's all I need, right?
If there's a moment where you have to sequence a salamander genome, you have to make Pete do it by himself. The entire project.
Oh,
Here's a club now.
Okay.
all right.
Questions.
Thanks.
Anybody else?
It's got 46 questions.
I think that's it.
I thought Margaret, and Richard, you were going to give me a hard time about my snide remarks around chemistry.
Well, we could point out that sodium ions are also positively charged.
Yes.
Sodium and potassium are both positively charged.
Oh, sorry. Yeah. Thank you. I need you.
All right. Well, thank you all for coming tonight. That was great, Tom. Thank you for doing that, especially on short notice.
You're welcome.
thank
It's
you.
fun. Glad y'all came by.
Very good to see you again.
Hi, Ceylon.
So
Steve.
good to see you again. Yeah.
Good to see both of you, such as it is.
Yeah.
It's good to see a lot of you folks. I haven't seen your faces. Stan, we'll get together.
Yes, June.
Okay. I've got it on my calendar.
That's the plan anyway.
All right.
Guys, next month.
Well, have a good night,
Bye.
everyone. We'll see
Pandemic
you next
willing.
month, December 11th.
Thanks, August, for putting this on.
Oh yeah, it's not just me. Thanks for coming.
Yeah, Pete's the one who told me about it. I didn't know. So thank you, Pete, for